Welcome back, everyone. Let's go ahead and move on to our next topic in Chapter 2 of Human Physiology. In the previous video, we talked about chemical reactions and enzymes. So the rest of this chapter is going to focus on some of the important macromolecules like proteins, which will be the last video here, and you'll have on your own work with nucleic acids, lipids, and carbohydrates. So let's use this video as a way to lay some of the fundamental groundwork for how some of those molecules are put together. So let's get into this. The first thing to understand is that living organisms are made up of both organic matter and inorganic matter. You might think of yourself as just purely organic matter, but you'll see that once we define these things that that's not the case. An organic molecule is one that contains both carbon and hydrogen at the same time. So, an example of this might be oil, right? So oils contain both carbon and hydrogen almost exclusively, so organic molecule. Fossil fuels are almost exclusively carbon and hydrogen, organic molecules. Coal and graphite and diamonds and things like that are just carbon, so they're not really organic molecules. They do contain carbon, but they don't also contain hydrogen. So what we're going to find out here is that even though the strict definition is that they have to contain both carbon and hydrogen, that does not mean that they can't contain other kinds of elements too. So it's going to be likely that a lot of our macromolecules and our organic molecules will also contain things like oxygen, nitrogen, phosphorus, sulfur, etc. So some examples here, I already laid out fossil fuels and oils and things like that tend to contain carbon and hydrogen. A lot of carbohydrates and lipids contain carbon, hydrogen, and oxygen. Proteins, we will come to find, contain carbon, hydrogen, oxygen, nitrogen, and sulfur. And then nucleic acids contain carbon, hydrogen, oxygen, nitrogen, and phosphorus. You'll notice the thing all of these have in common is that they all contain both carbon and hydrogen at the same time. Inorganic molecules, as the previous definition suggests, do not contain both carbon and hydrogen at the same time, although they may contain one of the two. Coal or graphite or diamond is literally just pure carbon. No hydrogen, so it's not organic. Water contains hydrogen but not carbon, so it's not organic. If something is going to be considered organic, it has to have both carbon and hydrogen. So the reason why we bring this up is because there are 4 classes of macromolecules that we are going to want to come to appreciate here. The most important, in my view, is going to be proteins. That's why I am going to dedicate a whole video in this chapter to protein structure and function. The others are still important, but I think having a full-blown discussion on them is less necessary, so that's why I am treating them as an on-your-own assignment. Carbohydrates, lipids, and nucleic acids, just make sure that you are paying attention to how they're constructed out of their monomeric components and what their general physiological function is. Again, these are all very large polymers that are built by linking an indefinite number of individual and small monomers together by dehydration synthesis, which we discussed in the previous video. You can see the diagram here again. So all of these molecules are built by using dehydration synthesis. The identity of the monomer varies from 1 to 2 or 2 to 3 or 3 to 4, but the concept of using the dehydration synthesis is still the same. All right, so let's talk about the unique chemistry of carbon atoms. Something that you got some practice with earlier in the semester with your on your own work is figuring out for a given element how many valence electrons does that atom have. So sodium has 1, lithium has 1, potassium has 1, oxygen has 6, sulfur has 6, fluoride has 7, neon has 8, so you got a good amount of practice figuring out how many valence electrons atoms have. Well, if you look at carbon, you're going to notice that carbon has 4 valence electrons. So if you're trying to figure out how to satisfy carbon via the octet rule, there's not really an obvious way to do it. It's going to be just as likely that it would pick up 4 electrons than it would lose 4 electrons. It's going to have an equally difficult time doing both. And as such, carbon is not ever going to form ionic bonds. Ionic bonds are really just going to be in cases in which atoms can easily transfer electrons they don't want or pick up electrons that they do want, in the case of, say, sodium and chlorine. So what carbon is going to do instead to satisfy its octet requirement is it's going to form covalent bonds. As such, it's actually going to usually form a total of 4 covalent bonds, meaning it shares a total of 8 electrons, and that satisfies its valence shell. So this is very interesting because this means that each carbon atom in a molecule can covalently bind up to 4 atoms simultaneously at the same time, and this also includes other carbon atoms. So the interesting part of this is the idea that carbon can bind to carbon, and oftentimes what you'll get is the production of a long chain of carbon atoms, and we call this a carbon skeleton. In this case, we can see we've got 7 carbon atoms chained together with covalent bonds, and each carbon atom also is going to form an extra 3 covalent bonds if it's on the outside, or 2 if it's on the inside, because these inside ones have 2 covalent bonds with other carbons. So for right now, it doesn't really matter what these atoms are going to be here on the outside of the skeleton. It could be just about anything. Not anything, but a lot of different things combined to carbon. So for now, let's think of these as vacancies. So these are just empty spots where carbon can potentially form a covalent bond with some other atom. It could be another carbon atom, it could be oxygen, it could be nitrogen, it could be a lot of different things, right? So for now, let's just think about this in terms of these vacancies. So the internal carbons each have 2 vacancies, and the carbons on the ends will have 3 vacancies. Well, first, let's consider what if we fill those vacancies with hydrogen atoms. You're going to see that that's really the simplest way to fill those vacancies. So doing this will yield a kind of molecule called a hydrocarbon. This is a type of organic molecule that contains just carbon and hydrogen. So the molecule that you see here is called heptane. Heptane is going to be a kind of fossil fuel, just like octane or propane or butane or some of the other fossil fuels you're aware of. So the lesson here is that heptane is going to be a very, very, very hydrophobic molecule. So if you kind of put this into perspective based on one of our earlier discussions on hydrophobic and hydrophilic molecules, you can see that because the carbon skeleton is littered with hydrogen atoms, and because these are all non-polar covalent bonds, if water were to try to surround this heptane molecule, water would not be able to form hydrogen bonds with it, so the molecule in and of itself is going to be hydrophobic. It's not going to like interacting with water because every single covalent bond surrounding the carbon skeleton is non-polar, and water is a very polar molecule, you will recall. Okay, so generally speaking, if you surround a carbon skeleton with nothing but hydrogen, you're going to end up with something that's just super duper duper hydrophobic. It's not going to want anything to do with water. Well, you can actually fill those vacancies with things other than hydrogen atoms. The other things are what we call functional groups, and functional groups are just these small chemical structures that can attach to carbon atoms, and they will impart to the molecule as a whole special chemical properties. Some of them are polar, some of them are non-polar, some of them have electrical charge. So let's take a look at what some of these functional groups actually look like. There are quite a lot of different functional groups that we could talk about, but this kind of covers the most important ones. First, you have the hydroxyl group, which is an OH group. Because you have an oxygen here bound to a hydrogen, this is going to be exceedingly polar, so if you want a molecule to be hydrophilic and interact with water, you probably want that molecule to have some hydroxyl groups. That allows the molecule, the organic molecule, to interact with water. Carboxylic acid groups do sort of the same thing, except we're going to find that these are going to be negatively charged. They act as acids and will give up that hydrogen atom, which causes the oxygen here to be negatively charged. Amino groups, or amine groups, as I prefer to call them, are also going to be polar, and we'll find that they tend to be positively charged at a neutral pH. Methyl groups are going to be very nonpolar. It's basically the same thing as just filling the vacancies with hydrogen, because everything you see here involves nonpolar bonds. And then the phosphate group is going to be another negatively charged one, and it will be very polar as well. Okay, so these are just some of those functional groups that you could find on a large organic molecule, but what I want us to be able to do, the skill I want us to be able to develop, is to just be able to look at a carbon skeleton and the functional groups that surround it and try to predict whether that molecule is going to be hydrophilic or hydrophobic. I promise you this is going to be a very important skill that you need to develop quickly here. So if we were going to take that hydrocarbon from before, where all the vacancies were filled with hydrogen, if we start plucking off those hydrogen atoms and filling them with, say, hydroxyl functional groups, that organic molecule is going to start progressively becoming more and more hydrophilic, because those hydrophilic groups are very polar, and the more hydroxyl groups you add, the more propensity you give to that organic molecule to interact with and hydrogen bond with water that surrounds it. Okay, so let's look at a specific example here. I took that heptane molecule from before, I plucked off 4 of the hydrogens, and I replaced those hydrogens with 4 different functional groups. So I want you to be able to see visually here the implication for these functional groups and what it means for this molecule as a whole's solubility in water. So let's cover each of these functional groups one by one. The first one that you see here is the amine functional group. It's polar, but actually at a neutral pH value, it is going to take on an extra hydrogen. It acts as a base, and it is going to take on a positive electrical charge. So polarity involves partial negative and partial positive charges. This is going to be a whole positive charge, so that's very interesting. So that will interact very strongly with water, just the same way sodium and chloride interact with water, right? The hydroxyl group you see right here, it does not have electrical charge, but it is very, very polar, so water will kind of congregate around that hydroxyl group as well. Over here on the end, you have a carboxyl or carboxylic acid group. The opposite of what we saw with the amine, it will want to give up its hydrogen atom. In fact, I drew this as if it already did, and that oxygen there is going to be negatively charged as long as we're operating at a near neutral pH. And then finally, the phosphate group down here will have a double negative charge, one here and one here, so it will be negatively charged at most pH values. So the way to kind of think about this is that if you picture this molecule being surrounded by water, you actually have several different places where water can kind of latch onto and solubilize the molecule as a whole. Water can surround the phosphate, water can surround the amine, water can surround the hydroxyl, and water can surround the carboxyl groups, because polar molecules like to congregate around other polar molecules. Now, these hydrogens are still going to give this molecule a little bit of hydrophobic properties, but there is no doubting that this molecule that you see here is much, much, much more hydrophilic than the heptane molecule that had nothing but hydrogen surrounding the carbon skeleton, of course. So the bottom line here is that the more hydrophilic functional groups you find on a carbon skeleton, the more soluble that molecule is going to be in water. And like I said, being able to make these sorts of predictions is going to be a very important skill for us as we move on later in the semester. All right, that's going to do it for this video. Thank you again for your attention. Join us for one more video in this chapter, and we will talk about protein structure and protein function. And as such, we will also circle back and have a little bit more to say about enzymes, since most enzymes are in and of themselves proteins too. So thanks again, and